Good morning. I am Dr.G.Vidhya, Assistant Professor, Department of Zoology, DKN College for Women. Today, I am going to briefly explain about the tools of biotechnology. What are all the important tools of biotechnology? And they are enzymes, cloning vector, competent host. There are several enzymes used in the biotechnology and they are lysozymes, DNA ligase, DNA polymerase, alkaline phosphatase, reverse transcriptase, nuclease. This nuclease enzyme we are going to concentrate little more deeper. What is the function of this enzyme nuclease? It's the enzyme which help to cut the phosphodiester bond of polynucleotide chain. See this diagram. This is the region where cut by this enzyme. The bond which present between the phosphate and the sugar group. And this enzyme are two types. One is exonuclease and another one is endonuclease. From the name itself we can able to identify which part they are going to cut. Exonuclease is an enzyme, they cut the nucleotide from the end of the DNA strand. Whereas in case of endonuclease, they cut the nucleotide from within the DNA strand. This diagram represents this enzyme cutting. So, this endonuclease is an enzyme, it's also called it as the reverse transcriptase and also called it as the molecular scissor. Actually, this is an enzyme which present naturally in the bacteria which act as a defense against the pathogenic virus. Okay, and this enzyme, how it defends against the virus? By cutting the viral DNA into the fragment. The function of this restriction enzyme is they go and bind the particular site, they go and bind the find the recognized site where they cut the viral DNA into fragment. So they are also called restriction enzyme, they also called it as the molecular scissor. So already I told about the what is recognition site. Recognition site is nothing but how the restriction enzyme go and recognize the particular site for cut that DNA material. So the specific sequence of the nucleotide, the restriction enzyme, they go and identify the particular site and they cut that site. And usually this recognition site is generally it's a palindrome. What is meant by palindrome? Palindrome, it's nothing but the DNA in which the 5' to 3' base pair sequence is identical on the both sides. So they also called it as the inverted repeat palindrome. See this diagram, GAATTC from 5' to 3', the same thing which present in the opposite stand also. GAATTC. Now we'll see how this restriction enzyme cut the particular site, that is recognition site. What is the end product of that cutting? This cutting is called it as the cleavage pattern. So they produce two type of patterns, sticky end fragment pattern and another one is the blunt end fragment pattern. See this diagram. In this, if the cut is given here in one stand, but in the opposite stand, it's not exactly to that of the first stand. So that resulted in this type of fragment. See here they have a overhanging of the sequence is there and also unpaired nitrogenous base is also seen. So this type of fragment, it's called it as the sticky end fragment. And coming to the next one, blunt end fragment. In this, the cut is given in this region. That second cut is also straight to that of the first cut. So they produce this type of fragment. Here there is a no hanging of the nitrogenous base and no nitrogenous non-sequence region. So these are the example, these are the different restriction enzymes and their mode of action. See, this is a restriction enzyme which is isolated from the Orthobacter luteus. This first letter represents the genus and second two letters are represent the species. And this number 1 is represent the first restriction enzyme isolated from this bacteria. So this produces the blunt end. See the this restriction enzyme cut the straight cut in the straight format, so they produce the blunt end fragment. Whereas in case of HI, that is Bacillus amphilolusiferians, okay, the same thing, but here they produce the staggered cut, so that resulted in the sticky end fragments. So coming to the next one, cloning vector. This is a second important tool. What is the cloning vector? Cloning vector, it's nothing but the which act as a vehicle which help to carry the gene of interest or the desired gene into a host for further multiplication. So when the vector replicates inside that host cell, obviously that gene of interest is also replicate. So this is a diagram. This is a foreign gene. Once it inserted into the cloning vector, this is a recombinant DNA. After recombinant DNA, we are going to insert it into the host cell. Inside the host cell, they multiply. So how we have to select that particular cloning vector? It should have some of the important characteristics. First important characteristics is it should have a ORI site. What does mean by ORI site? ORI site, it's nothing but the origin of replication. In that site only initiates the replication, and not only initiate the replication, it also control the copying number of the linked DNA. This is the first important thing. And second important thing is it should have a selectable marker. Why it should have a selectable marker? Then only we can able to identify whether that particular gene, that desired gene, is present or not. So usually what they use now? They use ampicillin and tetracycline. And third thing is the vector should have a recognition site or unique recognition site or cleavage site. So then only the restriction enzyme go and cut that particular site. This. Other than this, the cloning vector should be small and short, and it more compatible with the hostess and incompatible with the other vectors. These are the things, and also the cloning vector should move under both the systems, that is prokaryotic system as well as the eukaryotic system. These are the important characteristics of the cloning vector. So these are the different types of the vector: plasmid, bacteriophage, cosmic, pagemic, BAC, YAC, MAC, transposons, and baculovirus. So the third thing is the competent host. You all know about the DNA, they are hydrophilic in nature. So it cannot enter into the cell directly. So we have to make that cell competent. Then only it takes up the DNA. This process that is called as the competent host. For that, what they do, we have to treat that with the divalent ions like calcium, so that it helps to increase the pores of the cell, so the bacteria, the DNA easily enter into the host cell. And another method is micro-injection method. Directly the DNA inserted directly into the cell. Particularly what animal cells are follow this method. In plant cell, they usually follow the biolistic or biogene method. What they'll do, the cells are bombarded with the high velocity of DNA coated gold and then gold. So these are the competent things. Other competent hosts of bacteria are Bacillus and Helicobacter pyrrole and Agrobacterium tumefians. So today we understand about the tools of the biotechnology. Thank you.